This lesson is on the second part of section 5.2, which is active transport. So, in the previous video, the question was, what happens when we need to move materials from a high to a low concentration? And that happened naturally. The cell didn't have to expend any energy to make that happen. Well, what happens if the cell needs to do the opposite? What if it needs to move material from areas where the concentration is already low to a concentration where there's already more of it? So from a low to a high movement there in terms of concentration. So cells must use energy to do this. This is going to cost energy. We say this is moving up the concentration gradient. Okay, it's going against the natural flow here. Okay, previously in the last video we talked about how it's moving down the concentration gradient. This is moving up. This is not energetically favorable. So this is going to cost energy to do this. So the cells better have good reason for doing this. It's not just something they're doing for fun. So, what do we see here? Well, cell membranes. We've talked about other types of things within the cell membrane, but there's one type here that's going to perform active transport, and these are called membrane pumps. Okay, they're a special type of protein. Pumps help move substances from low concentrations to high concentrations. Now, this does require energy. Okay, so this is not free here. It's not like passive transport, right? This is active, so it is going to require energy. In both carrier proteins and pumps. So these ones, the carrier proteins were passive. We saw those in the last video, and the pumps are active. Okay, here are the similarities, right? The molecule still binds to a specific receptor on one side of the membrane. The protein changes shape. Once the molecule binds, the protein transfers molecules through the phospholipid bilayer, right, through the membrane, and then the protein releases the molecule either on the inside or outside of the cell. The only difference is, right, in the first instance, the concentrations were going from high to low with carrier proteins, and now they're going from low to high with these pumps. And the pumps, because they're going from low to high, are going to require energy. And it's usually in the form of ATP, right, the energy currency within cells. So, we're going to talk specifically about one pump here, the sodium-potassium pump. Okay, sometimes you'll see it written like that. Sometimes you'll see it written like that. So those are just the chemical symbols for sodium and potassium. This membrane protein pumps sodium and potassium ions up their concentration gradients, so it's making both of them go up. It will pump them in opposite directions, so if sodium is going in, then potassium's going out, right? Okay, or if sodium's going out, then the potassium is coming in. They're going in opposite directions. Most cells will have a higher sodium concentration outside the cell and a higher potassium concentration inside the cell. So it's probably going to be more like this second situation. The pump works to maintain this balance. So it works to keep sending sodium out of the cell and taking in potassium. So how does it work? Okay, so imagine this. All right, this is your phospholipid bilayer. Okay, and you don't have to necessarily draw this entire diagram, but understanding how it works and understanding how it's going to use ATP to make this happen, and that we're going against the gradient, right? We're going up the gradient here, okay? That's what we really are focusing on. But the first instance here, all right, we've got a carrier protein, right? This is going to be a pump, really, specifically. And we've got two sodium ions already bound, and a third sodium ion comes in. Well, it doesn't automatically just pump this stuff back out of the cell. Okay, so yes, those fit nicely into that structure there. ATP comes along and donates off a phosphate group. Okay, so it loses, instead of adenosine triphosphate, it becomes adenosine diphosphate, okay, which is ADP. So ATP becomes ADP and donates off a phosphate. That phosphate addition, step 3 here, changes the shape of this protein and pumps the sodium out. So now it's officially out of the cell, right? It's moved out of the cell. We've gotten 3 sodium ions out of the cell. So we're increasing the sodium concentration outside the cell. Well, while that's happening, potassium ions are going to come and fit in this pump as well, and they are going to cause the release of that phosphate group, okay, and the protein changes shape, all right, and it opens back to the inside, and then you have potassium being pumped right into the cell. So the potassium concentration is going up on the inside. Notice we got 2 potassiums going in and 3 sodium ions going out, which means that overall we're actually building more positive charge outside of the cell. So the longer this runs for, the more positive charge we get out, because there are 3 positive charges coming out for every 2 positive charges coming back in. So the cell is actually creating this gradient through this sodium-potassium pump. This is a video, won't load here in this app, but we will watch it in class so you can see actually it work in action. Okay, so just kind of some summaries of it. 1 complete cycle, you got 3 sodium ions pumped out and 2 sodium ions pumped into the cell. So how fast does this thing work? Here's kind of the cool part about this. At top speed, about 450 sodium ions and 300 potassium ions are moving around there. So you have 450 sodium ions moving out, 300 potassium ions moving in. So if you divide that by 2 for the potassium ions, that means this thing is working about 150 times a second, right? That's about how fast that pump is working when it needs to work at that optimal speed. As I said kind of at the end of that other slide, it creates an electrical gradient. We build up positive charge outside of the cell, and it's not really necessarily negative charge, but there's less positive charge inside the cell, so there is a difference in charge there. Okay, this is important in electrical signals of nerve cells. So like, where are these? I mean, a lot of cells will have these sodium-potassium pumps, but particularly for you, where you're going to find them a lot is on nerve cells. It needs sodium and potassium to signal that message. Okay, so when one cell needs to signal that it's sensing something, okay, that change in electrical signals there, having positive charge or not as much positive charge, sends a message to the next nerve cell, which relays that all the way back to your brain. And this is why things like Gatorade, right, and they say it's rich in electrolytes. Well, it's rich in sodium ions and potassium ions. And so what does that do if you're an athlete? Well, it allows those nerve cells to keep functioning like they should. You don't run out of sodium or potassium. Or if you run out of it, then your muscles get kind of all messed up when your brain's trying to send signals to them. It's not able to respond, right? And then you get cramps. Okay, that's what's happening there, is that those sodium and potassium ions are not there in a high enough concentration, and your nerve cells aren't sure what signals to send. Okay, they get kind of messed up. Same reason they tell you to eat, like a banana if you're having cramps, is because a banana is rich in potassium ions, which is going to help reduce that cramping and get your nerve cells functioning back to normal. So you do need these sodium and potassium ions in your body for the nerve cells to function. This is only one example of a pump, but it's a pretty important one in your body. Okay, so other ways to move stuff in and out of cells that is active. And this is about, what if we have very large substances? What if a cell needs to basically engulf something or just expel something out of it that's very large? It's too big to go through the membrane. It's too big to go through by diffusion, by facilitated diffusion, by channels, by pumps. It's just, whatever molecule this thing is, it's large. Okay, so how do we do that? Well, we do it through either endocytosis, which is bringing stuff into the cell, or exocytosis, which is removing it from the cell. Cells resort to endocytosis and exocytosis to transport very large molecules. So if we end up with very, very large things, okay, they're going to use these two processes to move it either in or out. They're both very active. They all require energy, and you'll see how it works here in just a second. So endocytosis, the process by which a cell ingests, external fluid, macromolecules, large particles, and maybe even other cells. So if you're trying to remember which one is which, endocytosis is ingestion. You're bringing it into the cell. Okay, so if you're trying to remember, hopefully that'll keep it clear. Basically, you're engulfing it. It's kind of what it will look like. Materials enclosed in part of the cell's membrane. So you'll have your cell here. And let's say there's something out here that it needs to ingest. Well, what's going to happen is, as it approaches, it's going to more or less fuse with the cell's membrane, and just become part of it. It kind of gets ingested in. Or sometimes the cell will even more actively go around and engulf the whole thing. The enclosed membrane becomes a vesicle, so inside of the cell, we end up with this vesicle, and that's either going to confuse with like a lysosome to get digested or other membrane-bound organelles, okay? We'll see kind of what that looks like here on the next slide with maybe a little bit better artistry. Okay, so here's our cell, right? Something needs to get into it. Here's the plasma membrane. All right, basically it just kind of pinches around it, okay, and forms a vesicle which traps this stuff. Okay, again, this is endocytosis. We're bringing it into the cell. So these things here, whatever they are, they're too big to go through the membrane. They don't go through a carrier protein. They don't go through an ion channel. They don't go through some kind of a pump. They're just too large, so the cell has to kind of engulf it using its membrane. So, what kinds of stuff do they have to do this to? Sometimes sometimes it's fluids that they need to get in or out, and so if it's liquids, in terms of endocytosis, this is called Pinocytosis. This is transporting of solutes or fluids. So, if you need to remember that, this is often thought of as cell drinking. It's drinking large quantities of liquid. And then we have phagocytosis. This is the movement of large particles or whole cells. A lot of organisms feed this way. They more or less just ingest their prey. They kind of swallow up around it and engulf it in. Right? It's too big to fit through a membrane or any type of channel or carrier protein or anything like that. You just engulf it. In fact, there's a whole list of cells called phagocytes. These are cells that ingest bacteria and viruses that invade an organism. Basically, the lysosomes fuse with vesicles full of bacteria or viruses, and then they get released. Okay, so the opposite process would be exocytosis. All right, this is going to be exiting the cell. Right, exocytosis is exiting the cell. Process by which a substance is released from the cell through a vesicle that transports the substance to the cell surface and then fuses with the membrane to release the substance. Essentially, it's the reverse process of endocytosis. You have some kind of a vesicle within the cell that moves outward towards the cell membrane, fuses with the membrane, and expels the content out into the environment. Right, releases the content to the external environment. Things that it might release: proteins, perhaps proteins needed somewhere else. If it's a multicellular organism, waste, very large wastes that the cell can't just break down or get rid of, or toxins. Again, things that can't break down normally, let's just get those right out of the cell by exocytosis. Okay. So where we see stuff like this, cells in the nervous system, the endocrine system, things that need to have very quick response times and send out messages, they're going to use exocytosis. All right, so what does this look like? Right, here's your vesicle that's either got some kind of a waste or some kind of a protein that needs to get outside of the cell, moves towards the cell membrane, it fuses with the plasma membrane, and then releases the contents out of the cell, right? So here's like an actual, a microscope picture of it. Okay, the vesicle is going to just release all of that content out into the environment. And this is another video about phagocytes in action. We'll watch this one in class together.